Hello, this is Alex Aljetz doing my biweekly for the week, 2 weeks of May 31st. I guess I haven't done one of these since summer, so it could like kind of double for more than that, but I didn't do a whole lot of work over the break since school. Main thing that I'm working on right now is helping Joelle with the OTP, um, machine learning model kind of stuff. So first I was, doing this a while ago, um, comparing the different assays, the assay descriptions on Kemble, trying to um, get trends out of it, look at it. There was this McFeely paper um, from Washington that kind of looked at the same kind of assay data, um, correlation between inhibition and the pre-incubation conditions, assay cell type, substrate a little bit. Um, so tried to replicate this project or this paper. Didn't really work that well with the Kemble data. Um, as long as my code was right, I'm pretty sure it was. Like I was getting all signs point to it's good. Um, Filtered it for per victim pairs that had over 3 experimental results. Um, no cases of that. I actually got 2, but it seemed like it was just the same 2 papers kind of twice. Um, so I emailed the PI lady on that paper, asking for access to the data set very politely. She didn't respond, so that kind of sucks. I know McFeely isn't at Washington anymore. I guess I could try to um, email her, and then I think there's like a lab manager or something like that that's also listed on the paper, who's still at Washington. Um, so kind of jumping off from that a little bit, I think. Um, I was graphing the results of the machine learning model, making heat maps with the different sampling methods, accuracy, precision recall, balanced accuracy, and F1, which there seemed to be more of a focus on balanced accuracy because it is all of them put together. Um, and then made bar graphs specifically for balanced accuracy for the best, you know, confirmation, feature used. There's like so many variables, it's a bit hard to like think about, but once I look at the graph, I can tell what's being plotted. Then wrote like a little figure caption, like kind of sample for each type of thing. This is like an example of the heat map. We looked at these in the OATP meeting. I don't think I need to spend like too much time on here. Um, then this is the bar graph, which I think is a little bit more insightful, where it's like, Plip and Rosetta for in and out is like the best, and using random undersampling. It's kind of like a good combination of balanced accuracy and a standard deviation. Um, Graphing more iterations of these. Um, Joelle sent me the hyperparameter optimized model. I'm pretty sure that's the model, the results that she sent me. Didn't seem like there are a lot of changes. Um, I put all the graphs in the research updates folder. It says like 530 on it. Um, it's just like the same as these guys. Um, Also, I'm comparing variability across specifically Carlin, Lane, and De Bruin databases for the manuscript. Um, got the raw agreement between them. I think it's like 70%, maybe 90% for one of them. I forget which is the 90% one. Um, and then I also found like the R squared values and did a Wilcoxon test between Carlin and Lane, which basically said that they were like significantly different. I didn't do it between Carlgren and De Bruin because De Bruin is it's like a percent of control, so it's kind of like an activity percentage, um, which Mercer told me I need to change, but I'll talk about that later. Um, so the R squared of Lane and Carlgren is like 61.6, but obviously there aren't a bunch of results. And then De Bruin and Carlgren is 2%. It sucks. And the other one didn't give me an R squared value. De Bruin and Lane. So that wasn't good. Um, so I did all that. Don't really know what else to do. In terms of statistics, I've asked ChatGPT lots of things. I have like a couple, like maybe this will be good, but it seems like if you look more into the stat, it's like, oh, this isn't meant for like my kind of data. Um, because ChatGPT is just like kind of spitballing based on my bad prompts. But yeah, Mercer said to convert the activity percentage to an inhibition percentage for De Bruin, which I didn't think of doing. I did catch that the De Bruin was the percentage of control transport, and I like factored it in, but I kind of just figured that the correlation would be negative, and I didn't really think of like converting it to inhibition percentage for to make it easier. And then obviously, if you feed it into the model, it needs to be an inhibition percentage, which is a point that Mercer brought up that I didn't think about at all. Also doing some wet lab stuff, making proteins, um, growing E. coli colonies. They are really good. I didn't put the picture of my colonies in this presentation, but I sent it to you. Um, on Wednesday, made the broth because we didn't have the broth with kanamycin or plates with kanamycin. Performed the T7 transformation on Thursday. Friday morning, the colonies are doing well, and then Monday, I'll make the saturated culture, scoop a little bit out of my plate. Um, do some protein production. Yeah, and then I'm kind of just getting more comfortable in the lab. I'm like, oh, I can follow instructions, but then I get to something I like don't know how to work it, don't know how to like wrap the plates up. Um, so I just kind of need to get familiar, I think. It kind of makes me feel dumb, but I do appreciate that I'm definitely learning stuff. And then just doing a little bit of writing, got references for the OTP paper, looked through it. And then did more kind of writing on the OTP paper on Friday. Well, I didn't really write, kind of looked for more critiques because I don't really know a whole bunch about the project, honestly. So I definitely wouldn't feel comfortable writing about it, at least right now, with the knowledge that I have about it. And I feel like I'm a little not confident in like critiquing writing because I don't know, I'm not really sure what is clear and what isn't and what we're trying to do and what's like bad for what we're trying to do. So I'm kind of still getting familiar with kind of what the paper is trying to do, I think. Because I've like critiqued people's papers before, but it's like writing about like books and novels and stuff. So a different kind of critiquing that I'm used to doing. Um, but I am trying my best, and I'm definitely going to try to be more like purposeful when I go and read scientific papers now to see what they do that's like presents information in a clear way, or like when it's okay to kind of get down in the weeds and stuff. But yeah, getting more experience with that. Um, and then now, or with the kind of Alzheimer's project, the variants and stuff, I have not, or I didn't do any progress on that. Um, but oh shit. Um, Basically I need inward and outward facing structures. So I definitely need to talk to Theo about that because I don't know what features to play with to try and get out an outward facing structure. I don't know that yet. Definitely need to talk to Theo. So that's what I got for this. I think I'm making good progress. Definitely feeling like the graphs are going well and stuff. Um, yeah, that's what I got.